In this video, I'll be discussing mRNA stability. First, I'll go over some background about what it is exactly, the pathways of degradation, and some terminology and experimental procedures we should know. And then we'll get into some experimental history with some specific examples. So first, let's take a look at a quick overview of what mRNA stability entails. So you can see up here, this is just our normal mRNA, right? You see it's fully processed, right? There's a G prime, a 5 prime G cap, and there's also a poly A tail, right? This is great. This is exactly what mRNA should be after it's fully processed, right? But then when we want to, let's say, break it down, right? We have a few pathways we can do. One that we'll be covering first is called an endonuclease cleavage, which is basically you cut it in the middle, right, or somewhere in the middle, and then basically you now get two halves. And the key thing about mRNAs is that you need both a cap and a tail. If you lose either, you get degradation. And you can kind of see the two pathways there. Down here, you can see this 3' to 5' exonuclease decay. This is when you have no tail, right? And so little Pac-Man, the little deadenylase, exonuclease comes and starts munching at his butt, right? On the other hand, if you decap, right, if you have no more cap, you get 5' to 3' exonuclease decay, and you get muncher down from the 5' end, right? And so basically, when you have endonuclease cleavage, both happen, right? Because then you have basically one part of the mRNA that has no poly A tail, and the other one has no cap. And so you just end up getting, this one and this one over here, right? That's just like an overview of mRNA stability, probably nothing new to you. But now we want to take a look at some specific examples. So one is the transferrin receptor. What this basically does, that's also called TFR, by the way, is it's just like a receptor protein that imports iron into the cell, right? Just think of like a transport protein. On the other hand, we have ferritin, and ferritin it can form a complex that basically stores iron and releases it in a controlled way. So the reason why this is so important is because iron, free iron at least, is actually very toxic to cells, and so there's two mechanisms of preventing our cells from getting damage from this. One is to not import too much at once, because if you already have a lot of iron, you probably don't want more iron in your cell, right? So that's kind of what the transferrin receptor can help do. Likewise, if you don't have enough iron, remember cells need iron, the body needs iron, right? It can just import it, right? And then ferritin, right? Let's say you have a lot of iron in your body and in your cells. Well, you can produce ferritin, and then it basically just stores it in a safe way, right? And likewise, if there's low iron, right, then ferritin should start releasing iron, which basically is just lack of ferritin expression, because we shouldn't be producing more ferritin if we don't want to store more, right? So I'll write that here. So low iron, right? We want more TFR and less ferritin, right? Whereas in high iron, we want lower TFR, right? Because we don't want more free iron, and we want higher ferritin, right? So this is sort of like reverse regulations, right? And we'll actually see for how these things are regulated. They're actually opposites, but they both concern mRNA stability. And before we do that, I just want to go over two more terminologies. One's called IRE, and the other one's IRE-BP. IRE stands for iron responsive element. This is just a segment of DNA that can bind to, that is controlled by iron, or something about it's controlled by iron. So imagine you have like a DNA here, right? Let's say in the presence of iron, right? This ends up regulating this sequence. And then IRE-BP, right? Remember, this is the IRE, is a binding protein that binds to this sequence. All right, so this is the IRE-BP, and typically iron binds to this, right, to regulate this sequence. And that's basically what we'll see. This is usually an mRNA, right? And then we have an IRE. This is our iron responsive element, right? And then IRE-BP will bind to it, or it won't, depending on whether or not it's bound to iron, right? And then whatever happens after is usually different. A common IRE-BP is called cytosolic aconitase, And this is very, very much in ferritin. Oh my god. Oops, sorry. Ferritin and a transferritin receptor regulation. Okay, so that's basically that's where this fits in the puzzle. Now, before we actually dive into the history, I just want to go over two very crucial experimental procedures that we need to know. So the first one is a nuclear run-on assay, and basically what this will help us to do is to determine gene transcription levels. Okay, so first I'll go over the procedure. Right, first you isolate your cells of interest. Right, we have our cell. Great, and then next we want to just isolate our oh my god. Okay, these steps are bad, but what we basically want to get is our isolated RNAs, okay? With our like RNA polymerases, they're chilling, right? And then they're gonna transcribe, right? They're gonna transcribe depending on how the state of the cell, right? If gene transcription is high for a certain gene, they're gonna be transcribing away, right? That's basically what we want to capture. And what we do is we stop this. But then we add labeled nucleotides, right? And then we let them proceed. So what this does is that if we have our RNA strand and we have a RNA polymerase, right? They're going to be transcribing, but a lot of it, or the remaining parts of it, will be labeled, okay? And what this, an important thing to note is that this prevents the methodology here. It prevents any new transcription from initiating. Okay, so you only get a continuation of a transcript. And then what you do is you isolate the RNA. Okay, isolation. And then you hybridize it to a labeled probe of your gene of interest. Right. So for example, let's say you're trying to assay for I don't know, insulin activity, right? Insulin transcription activity. Then you would get the insulin labeled probe, right? So imagine this is our hybridized probe with a little label at the end. This will associate with our RNA if it's there, right? That's sort of how this works. And then we just detect the radioactivity. Basically, what this does, if you really think about it, right, is a measure of RNA polymerase density. Because depending on how many of these transcripts we get is directly proportional to how much RNA polymerase we had, right? Because for every RNA polymerase, you should get, well, like about one strand, right? And each strand would be labeled after you add the labeled nucleotides. And so depending on how much activity of radioactivity you get, then that is perfectly what, that will be a proxy for how much gene transcription there was. Because more RNA polymerase somewhere usually means higher gene transcription in that place, right? That's sort of logical conclusion. Important thing to note, this probe just acts as like a scaffold. It doesn't actually have to be labeled. I don't know why I did that, but basically just makes it to where it will bind to a specific gene, right? So after you bind this, right, you end up washing away all the other RNAs, so you can just get the RNA of interest, right? Because think about it, you're gonna get a lot of random, like let's say this is gene A, and this is gene B. We only want gene A, right? We don't want gene B. And the way we actually differentiate that is using this probe, okay? That's a nuclear run-on assay. Next one, I would argue more important, is a northern blot. A northern blot, what you basically do, this is a way to, this is very similar to a western blot, except instead of with proteins, it's with RNA, right? So imagine a good motivation is that, let's say you have two different cells. Let's say you have an epithelial cell and then like a pancreatic cell, right? These guys have the exact same DNA, right? And let's say you don't test for proteins for some reason, and you want to test for the transcriptome, the RNAs present. They're going to have different RNAs. For example, the pancreatic cell will have RNAs for insulin, right? Whereas the epithelial cell, hopefully it doesn't, right? And so after you, I'll use these guys as an example, actually. So what you do is you first add formaldehyde, which will basically denatures RNA. Because remember, RNA can form secondary structures, right? This is what tRNA looks like, right? Give or take. And because there's a H bonding between these nucleotides, what formaldehyde will basically do is to, elongate it. formaldehyde. Oh, I can't. Oh my god, hide. Right, and so basically you just get linear pieces. This is very similar to like SDS, the function of SDS. Then you want to isolate your RNA, right? So that kind of makes sense. So you just have RNA on its own, and then you run gel electrophoresis on either agarose gel or typically polyacrylamide gel because you want it to be smaller, right? And so what you basically end up with is a little gel, right, with your RNAs on it, right? Now, what you do is you put a membrane on top. This is a membrane. And then you basically add a bunch of filter paper between these two. So on the top, you add dry filter paper. At the bottom, you add dry filter paper. And then you dunk all of this guy in a huge tank full of water. Or some sort of solute. Solvent, sorry, not solute. And what this will basically do is that once you submerge this guy, right, what's going to happen? Oh, and I'm sorry, you add a big weight on here. So what's going to end up happening is that you get capillary action of the water moving up the filter paper, right? Because think about osmosis, right? Water will go from a lot of wetness to a lot of dryness, right? And so what you basically get is a bunch of water moving upwards, right? And what this will do is it'll actually shift these RNAs onto this membrane. Okay, that's very important. This is all we're trying to do. We're just trying to get the RNA on the membrane, and this is what you end up getting. And then so what you get is your membrane full of RNA on it, right? And then you can visualize this RNA using X-rays. That's basically how it works. So you blast this guy with X-rays, and you can detect where the RNA is. That's basically Northern blot. In summary, is just a way to look at RNAs and distribute them in terms of size. So exact same thing, or very similar to Western blot, or just typical DNA gel electrophoresis. This is just a way for us to see the RNA, right? Okay, that was a lot of background. Now let's get into the actual transferrin examples, right? So first we're gonna look at the transferrin receptor. Remember, this is the TFR is transferrin receptor. Transferrin receptor. And remember, this will basically import iron into the cell. So when we have a lot of iron in the cell, right, we don't want more iron, so this should be downregulated. And likewise, and contrary to that, when you have low iron in the cell, you want more TFR expression, right? So what they did was they ran a nuclear run-on assay. Remember, this assay is for gene transcription. And they revealed that TFR transcription was not regulated by iron. So basically what this means is they added iron, they didn't add iron, they ran this nuclear run-on assay, and they saw the exact same amount of transcription. Okay? This just means, okay, this regulation is not at the transcription level. This is very contrary to a lot of the other mechanisms that we covered, right? Where it's a lot of transcription factors and like the lactoperon, right? Those are all at the level of transcription. This is not the same. Now, what they did was they ran Northern blot, okay? And Northern blot, they did that in the presence of iron and without iron. And basically, you can see the graph on the right. When there was no iron, you saw that, okay, the mRNA was pretty stable, okay? There was a lot of mRNA remaining. But when you added iron, there was a sharp drop in how much mRNA was there. Now, this, they calculated the half-lives of these. So when there was iron, it was about 45 minutes before half of the mRNA went away, okay? And in no iron, there was a 12+ hour half-life. This also just means they didn't find a half-life because it just didn't decay, right? And so this suggests that, okay, when you have a lot of iron, you actually get mRNA decay, right? The regulation is not at transcription level, it's at the degradation level, okay, of mRNA stability. So that's sort of what this suggested, but now we want to figure out what sequence or where on this mRNA, or maybe somewhere else, does this happen? So what they did, okay, up here you can see like the structure of your typical mRNA, right? You have a promoter, you have the 5' UTR, right? Then you have your open reading frame, and then you have your 3' UTR. They found that the 3' UTR was what was required, right? You can imagine that this is pretty easy to test for. You can just like truncate parts of these off, right? And then see how that is affected with our exact same analysis here, right? And then they also found that if you swap the promoter with another promoter, right, you don't get any effect on iron regulation. Like this thing doesn't change. It is still the exact same results, even if you have a different promoter. So this just kind of like further confirms this is not at the transcriptional level. This is at the post-transcriptional level, right? So what we know is that it's on the 3' UTR, and we've confirmed that it's not at the transcription level, but only after it's transcribed do we get this regulation. Now, what they found was after a bunch of mapping experiments, they found that the 700 nucleotide region that was required in the 3' UTR is just a bunch of stem loops like this, right? This is just a bunch of stem loops. There's 5 of them, and this is what was required for regulation. And something very similar was found on the ferritin mRNA, except this was at the 5' UTR instead of the 3' UTR, right? Except this was just one. But you can also see, that's pretty much what they found, right? So they now know, okay, we have a bunch of stem loops that are involved with regulation in both ferritin and transferritin receptor. Now, they identified that these sites were involved with stability, right? And so they found the instability and protective elements of each of these, or of just the transferrin receptor mRNA. Basically, what instability element is basically just a sequence that initiates degradation. In this case, it would be your iron responsive element, right? Iron responsive element. Because this, just like we identified up here, this is where iron can regulate mRNA stability, right? And so this is what we would call the IRE, or I guess this down here, right? So the IRE is the instability element because this is where instability can be induced. Now the protective element is basically what prevents it from degrading, right? And this is our IREBP, our IRE binding protein, right? Binding protein. And they ran a bunch of experiments where they induced mutations in both of these, right? So as soon as you induce a mutation, you block REBP binding. And basically, this is very, very, you see the results are very consistent with their model, right? Our wild type, obviously low iron, you want a lot of the receptor so you can get more iron coming in, right? So that's why it's high levels of TFR, right? When there's high iron, you don't want much iron coming in, right? So you have low. Now, when they induced the mutation, the protection element, right? If you look at the protection element, it would be right here, right? This is just saying the IREBP can no longer bind, right? And what you found was that you get low levels of both, suggesting that you get a lot of degradation, right? Degradation. So this suggests that, okay, so you need this IREBP to protect this site. Exactly why we call it a protection element, right? Which is great. And likewise, when they induce mutations in the instability element, right? So this IRE sequence, right? So that it could no longer bind to IREBP, we got high levels of TFR, right? Now this suggests that you need a certain type of RNA sequence, right, to have degradation. Because as soon as you change it, right, you lose your regulatory ability. This is constitutively expressed, right? Meaning that it's not being degraded. So you need a certain, you can't just mutate it around, right? So this suggests that both are very specific. You need this REBP, right? And you also need functional sequence for degradation to occur. So this was very key. Now, obviously, we want to see how this degradation works in the first place, right? And so what they did was they ran northern blot of just, okay, so what they did, you have mRNA, and then you're in, and you put it in a high iron condition. So remember, high iron should result in mRNA degradation, right? So you should start seeing less and less mRNA. And they did two types of experiments. They first did one where they only exposed it to a bit of X-rays, and then they did long exposure to X-rays. So basically what this does is when you have short X-rays, right, we only get significant bands. Significant bands. Whereas long exposure, you get very specific bands. But you don't get much differentiation, right? And so if you look to the right, in the short exposure, okay, you can see it slowly tapering off. This is great, right? This is exactly why we ran short exposure, because this confirms that, okay, the mRNA is slowly going away. All the other mRNAs down here, right, which there probably are, you just can't see because we didn't expose them to a lot of X-rays. Only the most significant and prominent bands will be there. And you can see it slowly tapering away. Now, at the long exposure, you can see this is a whole big mess, right? You don't really see much like decay or anything, right? Even these bands are actually mistakes, right? They're not meant to be there. That one is, but these guys aren't really necessarily meant to be there. Even this one. But what they did find was with long exposure, you can identify this little intermediate here, intermediate. Taken together, we see that, okay, mRNA getting degraded here, but then at the end, you get a little intermediate, right? And this is probably degradation product. And subsequent experiments confirm that this was an endonuclease cleavage. So this should make a lot of sense, right? We have our mRNA. We have our cleavage. Remember, this isn't high iron, so we have our IRE perfectly intact here with no binding protein on it. And you get your cleavage, and you get two pieces of RNA, and we're detecting one of them here. Since this is at the 3' UTR, this makes a lot of sense why it's very similar in size, right? Because you're actually just cutting off the 3' UTR. This is pretty similar in size to the original mRNA. You just kind of cut off the little tail there, right? Because it's already a 3' UTR. So this was very key. So now we have our final model of, okay, normally when there's low iron, we get binding of this IREBP. As soon as we get a lot of iron, it releases, and then we get RNAs cutting, right? Interestingly enough, for ferritin, okay, now I'm going to look at ferritin. Its transcription is a little reverse, right? If you remember, if you have a lot of iron in the cell, you want increased transcription, right? Whereas if you have low iron, you want decreased transcription. And so obviously the same mechanism wouldn't work, but the reason why is because it actually does something a little different, right? It still has the exact same IREBPs and IRE, right? We still have this figure works exactly well, just minus the RNAs here. Because instead, what we have is an AUG sequence here, or not AUG, AUG sequence here, where ribosomes are meant to go, right? A ribosome needs to bind here, but in low iron, right, when you don't need a lot of ferritin, it can't bind. It can't translate because it's being blocked by this IREBP, right? But then in high iron, this releases, and then you can get ribosome binding. And then he can translate. And this was at the 5' UTR. That makes a lot of sense, right? If it's at the 5' UTR, right, it's just going to start here, and then you can translate, right? It wouldn't really make sense if it was at the 3' UTR, right? Because the ribosome will just bind here, it'll translate it, and it's fine. So this makes a lot of sense. You can kind of match form and function, right? And so we can see the exact same mechanism, but very interestingly reverse results just based on what this IREBP ends up repressing. So that's pretty cool. But I want to end off with deadenylation, because this is another topic of mRNA instability, right? First, I want to introduce the RNase H assay. Basically, what they do is they take your mRNA and they hybridize, right? You can see here with a probe on a specific oligonucleotide, right? That should be in the region of the poly A tail. And what you do is you cleave with RNase H, which will only cut here at where this probe is. And so you can see that this is N, right? This is AN. This poly A tail could be any length. What this assay is doing is estimating how long each poly A tail is. And so you can see here, right, with a wild type cell, you can actually see that, yes, as time goes on, you see lower and lower lengths of a poly A tail, right? And you can actually see some bands increasing, right? Which is very interesting because as you decrease, right, as you're decreasing, you actually get a lot of little, single nucleotides or double nucleotides just chilling around after you cleave it off, right? And so you actually see increased concentrations of the smaller bands because you're cutting off the top ones, right? So that's pretty cool. And they identified deadenylase as the one doing this cleaving because you can see that when you have a deadenylase mutant, you get a big reduction in poly A tail degradation. And so yeah, this is DNA's activity, right? So this is just also a very key experimental procedure that they used to test mRNA degradation. And so to recap, what we did was we looked at this endonuclease cleavage, right? Remember, this was a transferrin receptor and ferritin mRNA, right? We also briefly covered a deadenylase here, and the rest over here should be isn't as important. I mean, it's all pretty similar anyway. Decapping would be interesting, but that wasn't really covered. But yeah, that's pretty much it. We covered how mRNA stability can be used as a regulatory interface to control for gene expression rather than transcriptional or translational level.